I'm Professor of Cancer Behavioural Science in the Centre for Cancer Screening Prevention and Early Diagnosis. I joined Peter Sassini's group at King's in 2019, and sort of since then I've been working quite closely with the Cancer Prevention Trials Unit, and then the whole team, as people will probably know, moved with the launching of the Centre for Cancer Screening Prevention and Early Diagnosis just over 2 years ago now. So I'm going to use this as an opportunity to highlight what's been really great about working so closely with the trials team in terms of the sort of work that we do. So probably most people will be familiar with the general principles of cancer screening, but just as a quick reminder, cancer screening is aiming to pick up cancer before it becomes symptomatic, so that we're picking up that early stage cancer where it's more amenable to treatment and outcomes are improved. And decisions about instigating new and changing existing screening programmes are made by an organisation called the UK National Screening Committee, and the UK NSC has a series of criteria for considering new and changing screening programmes. And I think this just illustrates the role that behavioural science has to make in this context. So there are criteria related to the condition, that it needs to be an important public health condition, that we need to understand something about its etiology, so we know that we can intervene at an earlier stage to improve outcomes, and that it's not amenable to other forms of prevention. The screening test itself needs to be acceptable, which again is what I'll come on to talk a little bit more about in a moment, as well as simple and safe. We need to have effective treatment for people that we pick up at screening, and the screening programme as a whole needs to be acceptable both to the people delivering it and the people who are on the receiving end. We need to weigh up the harms and benefits, and that might include psychological as well as physical harms, and we need to understand the opportunity costs of that programme. And then there are criteria for implementation, which include having adequate resources, both for delivering screening, for running good quality assurance, for following people up, and crucially to providing good information to those who are being invited. So broadly speaking, behavioural science can do these. It can do lots of different things, but it can do these three sort of steps, I suppose, in thinking about implementing new screening programs particularly. We can try to understand public awareness around new tests and new forms of screening, sort of information needs that are there, and to try and develop information to facilitate informed choice. We can look at acceptability of the actual test and the way that it's being delivered. Importantly, we can monitor and understand uptake and look at demographic and psychological predictors of uptake to look at targets for intervention, and also to try to ensure that screening is not widening inequalities. There's always the risk with any new health intervention that it gets taken up disproportionately by people who are at lowest risk, and then you get that widening of inequalities in outcomes. And then I mentioned earlier psychological harm. We can evaluate the harms and the benefits in terms of anxiety or relief or different psychological consequences of taking part in screening and receiving different results. And so these approaches can be applied to new screening tests that are coming down the line, which is what I'm going to be talking about mostly today, but also to changes to existing programs. So we've done work recently trying to understand what women might think about introducing AI into the reading of screening mammograms, to understand attitudes towards risk stratification in existing screening programs, and towards extending screening intervals where we've got improvements in the screening test, and we need to understand how best to communicate those sorts of changes. So hopefully that gives you a sort of snapshot of the range of different types of work behavioural science can contribute. And I should have said at the beginning, there's a really great wider team of behavioural scientists in CC SPED. So Laura Marlowe, Suzanne Scott, Samantha Quaife, and Georgia Black are all working in the sort of social science end of cancer screening and early detection. So this just gives you a little sense of the kinds of projects that the team is involved with. So I'm going to talk in a bit more detail about BEST4 and the NHS-Galleri trial, but people will have seen news about the launch of Transform last week, and Samantha Quaife is leading work on the behavioural aspects of that. That's the big prostate screening trial. I'm involved in the new EDIS trial, which is looking at doing a very large study to evaluate the role of AI in reading breast screening mammograms. We're working closely with Ranjit Manchanda on Protect-C and Detect-2, which are both genetic testing projects, and Protect-C particularly is interesting because that's looking at population-based screening for cancer mutations, so things like BRCA genes. So it will be really important to look at the psychological and communication aspects of that. And then there's also Improve, which is a CPTU, Cancer Prevention Trials Unit, project on prostate screening, which Ellie Mashopoulou is leading on, and Uscreen was an example of one that's now completed, but looking at HPV self-sampling and cervical screening. So a really wide range of different innovations in existing programs and also new tests coming down the line. So the work I'm going to talk a bit more in detail about today focuses on that psychological outcomes end of things. And the first example I was going to talk about is multi-cancer early detection test screening. So there's a lot of interest at the moment in whether we can use liquid biopsies, so blood samples, to look for markers of a range of different cancers. And in this Lancet Oncology thought piece, we tried to lay out what we thought the issues were in this context that behavioural science can really add value and understanding to. So there are some really unique features of NSAID screening which are different from single cancer programs. So it uses a very simple blood test, which could well be an advantage compared with some of the other screening tests that are out there at the moment. People are likely to find that a familiar procedure. Obviously, the key difference is it's screening for multiple different types of cancer in one test, and the knock-on effect of that is that the follow-up is very complex. So whereas with breast screening, it's possible when someone's making a decision about taking part to explain to them exactly what will happen following an abnormal result, whereas for an abnormal MSED result, there are a whole range of different pathways that people might find themselves on, depending on what the likely site of origin of that signal is. We also think that MSEDs are likely to have a higher specificity than existing cancer screening tests, so that has psychological implications for a positive result, in that that positive result is likely to be more indicative of cancer than, for example, an abnormal mammogram is at the moment. And then there's this really interesting psychological issue of lack of diagnostic resolution. So if someone has an abnormal mammogram and they potentially get referred on for a biopsy, they might find that what they've got is not cancer, but it's a cyst, and so that explains that finding. And similarly, if someone goes to their doctor with symptoms, the symptoms are investigated. Maybe they've got bowel symptoms that don't turn out to be cancer, but there's evidence of colitis. Again, you're getting a sort of diagnostic resolution and an explanation for what that symptom or screening result meant. Whereas at the moment, if someone has a cancer signal detected result from an MSED and cancer can't be found, they can't be given that alternative sort of explanation. And so this has implications for the kind of behavioural science that we can do in this context around trying to understand acceptability and likely uptake of an MSED blood-based screening programme, implications for how we develop optimal communication around results delivery, and also trying to understand those psychological impacts of a positive result. And we've been able to start doing this working very closely with the NHS-Galleri trial. So the application for this trial was put in when we were back at King's, and right from the beginning we were the behavioural scientists were involved in these discussions about integrating behavioural science and psychological outcomes into the plan for the trial from the start. So this is Grail funded trial looking at their Galleri test, and you can see the sort of trial design. So people attended a mobile unit like the one you can see on the left. They gave a blood sample. They were randomised into the test or control group. Those in the test group had their sample tested with the NHS-Galleri test. If a signal was detected, that was communicated with them, and they were followed up on the 2-week wait referral pathway. If no signal was detected, they weren't told their result, and everyone was then, if they hadn't got a cancer diagnosis in the meantime, people were invited back at 12 and 24 months. And the trial has been kind of amazing to watch the trials unit at work with this, but to have recruited 143,000 people in 10.5 months was an extraordinary feat of logistics, which was amazing to witness and be part of. And then within this, we've nested what we call the signal sub-study, so looking at the psychological impact of the Galleri test. Because people didn't get a negative result, we didn't think it really made sense to collect data from people in the negative or control group, but we've done surveys with all the people who got a cancer signal detected result in the trial, followed them up over 3 time points. And also nested in some qualitative interviews. So the main aims of the Signal project were to establish the levels of anxiety among participants with a cancer signal detected initially after they received that result, and then to compare longer-term anxiety at 6 and 12 months between those who went on to have a cancer diagnosis and those who didn't. And then we've got a nested qualitative study to do some in-depth interviews with people selected from those surveys. And you can read the protocol paper in BMJ Open, and we've also published the first of the qualitative analyses in the British Journal of Cancer earlier this year. So this is what happened to people. They had the blood test. If the cancer signal was detected, they got a phone call from a nurse to explain that result to them. And then this was followed up with a letter confirming the results. And our first questionnaire, the T1 questionnaire, went alongside those results in that letter. They were then referred on the 2-week wait referral pathway in line with wherever the cancer was thought to be. We then followed them up at 6 months, by which time they would either have a cancer diagnosis or had been told that cancer was not found. And then roughly at the 12-month anniversary of the blood test, we sent them a third questionnaire, and this was slightly tailored to whether they had had cancer diagnosed, they had attended for that second blood test, or they hadn't had cancer diagnosed, but they didn't attend for the blood test. So 3 versions of that. And you can see this is what the questionnaires looked like. And people were sent a paper copy, but also had the option to log on and complete the questions online if they preferred to do it that way. So our primary outcomes were, the primary outcome was anxiety, measured using the state trait anxiety inventory, which has been used across lots of different screening studies, so enables us, although we haven't got a comparison group with a normal result in this trial, we have got other comparators in different screening contexts. We used the cancer screening questionnaire to look at emotional, physical and social consequences of screening, and that includes both negative and positive subscales. And then again, for comparison with other screening studies, we used these single item measures of test-specific concern and test-specific reassurance, as well as changes to health behaviours, which was particularly people's responses to symptoms and intentions around participation in other screening programmes. There's been quite a lot of concern that if someone takes part in an MZ screening test, they might think that they don't need to take part in the other screening. So there's a range of outcomes here. Anxiety is a very general measure of anxiety. The other measures are digging a bit more specifically into people's responses to the actual result. And then within the second survey, we asked people if they would be willing to talk in more detail about their experiences, and then contacted some of those for an interview study. So I can't talk about the, although we've got all the data from the surveys and we're starting to look at it, Laura Marlowe's taking the lead on that analysis. I can report some findings from the qualitative study. So we did 41 interviews. We selected people to be roughly equally in the cancer diagnosed and the no cancer diagnosed group, so that we'd get a good understanding of both of those different experiences. You can see here that we had a good balance in terms of gender and age. As with the trial, we did fairly well. I mean, the trial did very well at over-sampling people from more deprived backgrounds, so that the distribution across deprivation quintiles was very even. We didn't do quite as well, we're slightly skewed towards the least deprived groups, but we've got good representation from all quintiles, mainly white British, and a range of levels of education. And the median time from receiving the result to having the interview was just over 7 months. So this gives a sort of overview of the study findings. And as I say, this is a paper that's published. So if you're interested to dig into this in more detail, you can have a look at the paper. But we described people's experiences along this pathway from receiving the signal detected result to having the diagnostic tests and then the diagnostic outcome. And across that pathway, people's expectations about cancer seem to be really important in determining how they would respond to those different stages. And there was a range of experiences. So frequently people expressed shock and surprise when they were diagnosed. They were taking part in research. I think people often don't think that might lead to an actual result that will have an impact on them. People tried to make sense of the result, and that was sometimes in terms of talking about the coherence of the predicted cancer signal origin site, so where the cancer was compared with things that were going on in their lives. So someone said they'd had a PSA test for prostate a couple of months earlier, but there was a bit of swelling, and so maybe then getting a prostate result was linked with that. For other people, it didn't make sense if they, for example, had a clear screening result recently. People sort of grappled with the implications for their cancer risk and also the social implications. So someone talked about the fact that they'd just heard they were going to be grandparents, and this made it worse because they were thinking, what if they weren't going to be there to see the baby being born? There was then concern about cancer at the point where they were having diagnostic tests, and often people felt a pressure to discuss that process. For some, it was quite drawn out. Laura's done some really nice work sort of showing the different kinds of timelines, but sometimes there are two cancer signal origins, and so people find themselves moving from one pathway into another and having to wait a long time for results. So that protracted process sometimes made people feel that they had to disclose what was going on, and then it was could be frustrating if then there was no cancer and they'd worried people as well as themselves. And then in terms of the diagnostic outcome, some people where cancer was diagnosed felt quite prepared. So they'd been through this process and sort of gradually realised that probably cancer was going to be the end point that they would reach. Whereas others, because of other, this expectations of cancer sort of aspect, got through that whole process and still felt very shocked when they got the cancer diagnosis at the end. Some people who had no cancer felt very reassured from having had investigations and felt that they were confident that there was no cancer. Others sort of grappled with what the result meant in that context. So someone said, I suppose there's a question in the back of my mind, okay, am I going to be developing a detectable cancer that they can't quite detect yet? So that idea that there might be something there that the test has picked up, but that the diagnostic workup hasn't managed to find. So from this work overall, there are obviously some quite striking similarities with having abnormal results across screening programs that are also found in this context, but also some novel aspects of MSED screening. So often the cancer signal detected result was interpreted in light of other screening test results, and people really found it a challenge making sense of the result if cancer ultimately wasn't found. But there was very clear variation even across 40 is a relatively small number of people that we'd interviewed, but we found a lot of variation in how people reacted and made sense of their results. And I think there are lots of good indicators in there about how we might do a good job of designing written information for people and sort of identifying people who might need additional support if the results of the trial are positive and if this sort of screening gets rolled out in the future. Just to mention the other aspect of this work that we did, which has been led by Ninian Schmeising-Barnes, which is outside the trial and which is trying to do, when I showed that diagram at the beginning, the sort of first bit about awareness and potential social inequalities. So we've also been doing this work to try and look at attitudes towards MSED blood test screening in the wider population who might be eligible if such screening was introduced, and particularly focusing on minoritized ethnic groups. So we've been trying to do that alongside the in-depth work within the trial. So the second example I wanted to talk about is BEST4, which is one that's ongoing. So Galleri, NHS-Galleri is now finished. BEST4 is ongoing. And again, we've managed to work really closely with the trials unit to integrate some behavioural science into this project. So BEST4 aims to evaluate this device called the cytosponge, people might be familiar with. It involves swallowing the little capsule that you can see on the right, attached to a thread. The capsule dissolves in the stomach, releasing the sponge that you can see on the left. And then as the sponge is retrieved, it takes a sample of cells from the length of the esophagus. It's been shown to be very effective at picking up Barrett's esophagus, which is a precursor to oesophageal adenocarcinoma. And this trial has been powered with a mortality endpoint to see whether screening with the cytosponge, or the capsule sponge test, as it's called, can be an effective way of improving mortality outcomes for esophageal cancer, which are pretty bleak at the moment. So the trial is recruiting women aged 65 to 79 and men slightly younger to reflect their risk from 55 to 79, and is targeting people with regular heartburn or reflux symptoms, or who take regular medication for heartburn and reflux, which is the group that's at elevated risk of Barrett's esophagus. It's being run as a trial within a cohort, so this just shows the sort of steps of the process. So it's a large population-based cohort with heartburn, and they're hoping to recruit around 240,000 people. So that is a bioresource called Heartburn Health. And then from that, when people are recruited into Heartburn Health, they give consent to be randomized into other studies, and so a Zelen design can be used so that only people in the intervention arm know that they've been randomized. Part of the enrollment for Heartburn Health involves watching a little video of the capsule sponge test and saying, if you were offered it, whether you would be likely to have it or not. And people who say that they would be willing to have it are then randomized 2-to-1 into the interventional control arm, and those 40,000 who are in the intervention arm are being invited to a mobile unit to have the capsule sponge test. And the hope is to screen about 26,000 people. So in collaboration with Emma Liddington, who's the co-PI on this project, I applied for some Cancer Research UK funding to try and understand the psychological impact of taking part in this kind of screening. So the aim of the screening is obviously initially to pick up Barrett's esophagus. This is something that puts people at elevated risk, but not at high risk of cancer, but that does need ongoing surveillance. And so there's a risk that you're creating a lot of anxiety and psychological harm in a large number of people, most of whom still won't get cancer. So we were keen to use the trial as a really good opportunity to collect some psychological data on this and to potentially understand people's concerns, so that if it were rolled out more widely, we could do a good job of trying to mitigate psychological harm. So a bit of a complicated diagram, but essentially we will be collecting, similar to the Galleri trial, data at 3 time points in the control group, as well as the normal and abnormal screen group and the non-attenders. So that first time point is around the time of receiving their results, or the equivalent for the control group. And then we'll be following up people about 6 and 12 months later. And at that second time point, people in the abnormal screening arm will have had a diagnostic endoscopy, and they'll know whether they've got Barrett's esophagus or potentially adenocarcinoma. And then again, we're going to be doing a nested interview study. So just with people who've had this abnormal screening result, we'll be interviewing them to find out in a bit more detail what that experience has been like for them. This is the conceptual kind of map of, we're using something called Leventhal's common sense model, which describes the way that people respond to symptoms or a label like Barrett's esophagus in both cognitive and emotional terms. So they try to make sense of that label or that illness by sort of creating a cognitive representation of what that means. And then there's also an emotional response, which might involve worry or might involve sort of denial or different ways of coping. And I won't go through all of this in detail in the interest of time, but we'll be collecting a whole range of these psychological measures. Again, our primary outcome is anxiety, to try and see whether we imagine quite sort of reasonably people who receive that abnormal sponge test result will be at elevated anxiety. We expect anxiety to be highest in those who have a dysplastic Barrett's or a cancer diagnosis. But in line with previous work, for example, in the context of HPV testing and cervical screening, we expect that anxiety to go down over time and to probably be returning to normal. So we will see how that goes. We've just managed to get these questionnaires going, again with huge support from the Cancer Prevention Trials Unit team. So people are being sent links to complete these surveys on REDCap, and we've got about 650 surveys back so far, out of a total of, I think we're hoping for more like 6,500. So lots a way to go on this, but really interesting and useful to be able to collect these data alongside the trial. So to conclude, I hope I've shown you something about the way that psychology and behavioural science can play a role in the development and the implementation of new screening innovations. The work that we do really aims to be able to help improve communication about screening, to ensure that uptake is equitable, and to make sure that psychological harm is given adequate consideration amongst all the other factors that are taken into account when deciding about new screening programmes. We know that the UK National Screening Programme recognises the importance of acceptability, and we're keen to help to add to that evidence base. And I will just finish by acknowledging all the teams that have been involved in these two studies, funding from GRAIL and from Cancer Research UK, and that's the wider CC SPED group at our away day last year. Thank you very much.